Another relatively new way to molecularly identify species that's possible because of high throughput sequencing is this approach called metagenomics. And what's unique about metagenomics is that it allows you to get large amounts of sequences from many different genes distributed across the individual's entire genome. So here is an example workflow. You might start with something like a fecal poop sample. So it's similar to metabarcoding in that you're still able to take this bulk sampling. So you don't have to separate out each individual separately. With metagenomics, you're able to do this bulk sampling. And because we're doing that bulk sampling, it's still necessary for us to do high throughput sequencing to get the sequence data back. So those are two commonalities between metagenomics and metabarcoding. One, we're doing bulk sampling. Two, we're still using high throughput sequencing. But there are differences between metagenomics and metabarcoding. So if you want, you can pause the video, try and think on your own, maybe look back at your notes, think about how this metagenomics technique is different from metabarcoding. And one of the main differences is that metagenomics does not use universal primers. So what it can do is give you information about multiple types of species at once. This figure I'm showing here is not mine. I have the credit listed here, but it's a super awesome figure, I think, that compares the techniques we've talked about so far and illustrates how they're different. So if we start at the top with DNA barcoding, we said you needed to get samples from each individual separately. So this particular individual was studying bats. So what they had to do was take a DNA sample from this individual bat, use PCR and amplify this one particular genetic barcode region to figure out what type of bat it was. For metabarcoding, if we keep moving down the line here, you're able to take this bulk sample. So now you're taking a bulk sample of insects using universal CO1 primers for PCR to amplify this barcode specific region across all insects. And then you're able to identify through high throughput sequencing these different species of insects that were present in your original bulk sample. With metagenomics, you're not doing that PCR step at all. So what that means, so if we look in metabarcoding, all of these samples are lined up because you're amplifying the same exact region, CO1 for instance, in all of your samples. With metagenomics, that PCR step isn't present, so you're getting these DNA samples across the entire genome. So you're getting sequencing from many, many different regions for all individuals that are present. So because you're not using universal primers, you're able to get information about multiple different species. So say we were sampling from bat feces, bat poop. If we use a metagenomics approach, we can figure out what bat it's from, what insects it was eating, and the bacteria and viruses that are present within the gut of that bat. So if we're using a metagenomics approach, we can get information for all of those species at once. So one way that I look at it is if you're talking about a technique that has the word barcoding in it, that means you're using one particular region of the genome. So barcoding, both of these steps use PCR to specifically target that one particular region, often CO1, that you're looking at. If you're talking about a technique that has the term meta in front of it, that means you're doing bulk sequencing in both cases and having to use high throughput sequencing. So meta, bulk sequencing and bulk sampling and high throughput sequencing. So those are some similarities and characteristics among the different types of techniques we talked about so far. But one of the main advantages of metagenomics is that you can get information about multiple types of species at once. And another main advantage of metagenomics is that because you're sampling or getting DNA sequence information from multiple regions of the genome at once, metagenomics can tell you not only what species are there, but also what those species are doing. So here in this workflow image, this is showing metagenomics, and at the bottom here, this is what I would like you to focus on. So just like we talked about on that previous slide with the bat feces, you can tell what different types of species are there. That's considered phylogenetic or taxonomic classification. So metagenomics can tell you what species are there. But because we're getting information from multiple different regions of the genomes for all of those species that are present, it can also tell us information about functional genes. So when we're doing barcoding techniques, we're often amplifying a region like CO1, for instance. That particular sequence region of CO1 can help us identify what species it is, but it doesn't tell us any information about really what types of metabolic pathways or what types of functional genes are present in that species. So if we're using the same example of bat poop, we might want to know not only what bacteria are present in the poop of that bat, but also what type of things those bacteria are doing. What type of processes they're doing. Are they breaking down certain types of metabolites or producing certain types of metabolites? And with metagenomics, we're able to get both types of information. What bacteria are there and also what they're doing, what functional pathways they are involved in. And this is actually an approach that we're doing in my research lab. And of course, when I say my lab, I mean my master's student, Emily. She's the one doing all of this work. But what we're using metagenomics for is to look at the relationship between the gut microbiome. So the gut microbiome is the community of bacteria and microbes that are present in the gut or intestinal tract. And we're looking at how the different community of that gut microbiome might be affecting thermal tolerance or ability to survive warm temperatures in red abalone. There's been some evidence from other organisms that if you have a more diverse microbiome, you might be able to survive warmer temperatures. That hasn't been looked at yet in red abalone, which is what Emily is focusing on. So for this study, what we're doing, or what Emily is doing, we're collecting feces from juvenile abalone, and what we're then doing is extracting DNA from that feces to be able to tell us information about that gut microbiome. And here I'll illustrate, again, some differences between metabarcoding and metagenomics to explain why we chose this metagenomics technique. So if we took the metabarcoding route, remember, if the term barcoding is there, that means you're only looking at one particular region. So metabarcoding would be able to tell us what bacteria are there. However, if we took a metagenomics approach, that will enable us to figure out who is there, so what bacteria are there, and what they're doing. And the reason why it's important for us to get this what are they doing information for our particular study is that one of the main ways previous studies have found that the gut microbiome affects thermal tolerance is that because of the unique functional genes that are present in these different types of bacteria, they're able to produce different compounds that help the host organism, like the abalone, for instance, survive these warm temperatures. So for our study, it's really important for us to know what those different types of bacteria are doing in terms of their functional pathways, what type of genes they might have that would allow them to break down or produce different types of compounds. And so for us, it was really important to choose this metagenomics approach because we needed that extra information of what the different bacteria within that gut community of the red abalone were doing. We would not be able to get that type of information with a metabarcoding approach.